Andraz Sturm, and I work at the Institute of Microbiology and Immunology at the Medicine faculty at the University of Ljubljana. I'm going to talk about our work about the simultaneous presence of different carbapenemase producing CPE within the same patients and the connections between plasmids for the transfer of the said genes. As we all know, plasmids play an important role in the genetic transfer between bacteria because they possess different antimicrobial resistance genes and virulence factors, which can cause transfer of these elements between different species within the same patients, maybe, for example, in the gut or at the site of infection, which makes treatment more difficult. It can also cause the uprise of multi-drug resistance bacteria, which is bad for the clinical setting. The aim of our study was to determine whether plasmid transfer could explain the different presence of different CPE species within the same patients producing the same carbapenemase. We had 18 patients with 2 or more CPE species harboring the carbapenemase genes for a total of 42 isolates, which were selected for long-read sequencing with GridION. The reads were assembled into genomes using FLY, and the plasmids were firstly assessed using SEQ-sphere to determine their lengths and replicant types. Only the plasmids with carbapenemase genes were selected for further analysis. PLING was used to identify the identities between plasmids and infer the clustering of the plasmids both within the same patient and throughout the whole cohort. What we found out was 3 different genus of bacteria, Escherichia coli, Klebsiella pneumoniae, and various different Citrobacter species. We found 4 different plasmid types, INK-LM, INK-X3, and INK-A and INK-N. The latter 2 contained the BLA-Vim gene, while the first one, the most abundant one, also present in the figure 2, harbored the BLA-OXA48 gene, and we can show the high clustering, high similarity of different plasmids between different patients, with a few of the plasmid within individual patients forming their own little groups, while still highly identical to others, but with some structural changes between them, mostly deletions and insertions. For analysis of the within patient plasmidome, we checked different species, and as you can see in picture 1, we found Citrobacter portucalensis, E. coli, and Klebsiella pneumoniae, all with exact same plasmid lengths, and according to PLING analysis, the plasmids were completely identical both in sequence and in terms of possible structural events. And the alternative visualization of identity was presented with the G-view. With this analysis, we showed high probability of intra-patient plasmid transfer between species. And for further analysis, we could check the temporal differences between the plasmids to determine whether it was actually conjugation or whether the plasmids were previously obtained by the bacteria independently of each other. But a challenge here is the use of only long-read data, which have lower accuracy than short-read, which makes the exact SNP-by-SNP basis discrimination more difficult. But that is time for further research. Our research also showed the potential of using PLING in larger scale plasmid outbreaks, which of multi-species outbreaks within the hospital, which could elucidate the transfer of resistance genes between different bacteria and different hospitals due to plasmid transfer and not actual bacterial outbreaks. In conclusion, we were able to successfully sequence different bacteria with long-read sequencing from different bacterial species in individual patients. And PLING analysis showed possible transfer of plasmids between these different bacteria. which highlights the potential for larger scale outbreak investigation in hospital in multiple species outbreaks. I would like to thank my colleagues who helped me work on this presentation, and also special thanks to ChemoMate and their Colibri project who helped fund this research.